Good afternoon, everyone. Welcome to the afternoon session on psychedelic drugs. My name is George Mashour, and I'm going to be moderating this session, but I'm also the first speaker. And I'm really excited to talk to you about two very interesting drugs, ketamine and nitrous oxide, which can serve both as anesthetics and psychedelics. And let me tell you why I think these drugs are interesting. On the one hand, we have the class of general anesthetics, and I've listed three here, two intravenous agents, propofol and etomidate, and also one of the inhaled anesthetics in common use, halogenated ethers. And I've referred to these as canonical anesthetics because they all work, to a greater or lesser degree, through the GABAergic neurotransmitter system, GABA being the primary inhibitory neurotransmitter in the brain. And as we've heard at this conference over the past few days, general anesthetics are wonderful tools to modulate and study the level of consciousness, the overall state of arousal of a creature, awake, alert, somnolent, sleeping, anesthetized, comatose, brain dead. On the other hand, we have the drug class psychedelics, and you've heard a lot today about the classical or the canonical psychedelics, psilocybin, LSD, dimethyltryptamine. And the reason I'm referring to these as canonical psychedelics is, just as the anesthetics that I've listed work through the GABAergic system, these canonical psychedelics are thought to work primarily through the serotonergic system. And of course, you heard a beautiful lecture on this today by Dr. Carhart Harris. Just as general anesthetics are effective tools to modulate the level of consciousness, so too are psychedelics wonderful tools to modulate the content of consciousness. Now, if you think about these two classes of drugs, anesthetics, which are used to suppress consciousness, psychedelics, which are used to enhance or expand consciousness, you wouldn't really think that there would be an interface, and yet there is. And sitting at the nexus of these two drug classes are ketamine and nitrous oxide. And this is something that I've focused on and that we focused on in our center for the past 5 years or so for two reasons. First of all, ketamine is neither a canonical anesthetic nor a canonical psychedelic, and yet it can have very similar traits depending on the dose. Similarly with nitrous oxide. And my thought is that if we can understand this outlier and how it connects with fundamental mechanisms, we can have a deeper understanding of how anesthetics work on the one hand or psychedelics work on the other hand. The other reason why I think these drugs are attractive, I talked about modulating level or content of consciousness, but you could use ketamine, for example, within a single protocol to explore a repertoire or range of conscious experiences, from baseline consciousness to a psychedelic state to an anesthetized state, all using the same drug depending on the dose. So that's the why for me of the interest here in these drugs. In terms of the how, at the Center for Consciousness Science at the University of Michigan Medical School, we explore these drugs and many others in many other states across the translational spectrum. Computational brain models, work in rodents, non-human primates, human volunteers, and patients, including surgical patients. And today I'm going to talk to you about some basic neurophysiology and neurochemistry from monkey and rodent work, as well as some of the neurophysiologic work in a clinical trial in humans. Now, there's a lot of great work out there on ketamine, and today, because of time limitations, I'm just going to be focusing on ours. So I'm going to start with the monkey, and what you're looking at in the top left of your screen is the surface of the monkey cortex with multi-electrode arrays overlaid upon them, and to the right you can see some of the neural activity that's recorded from these electrodes. So this can give us insight into neuronal spike activity or local field potentials, and I'll show you some of that. But the location of these electrodes allows us power to look at information transfer. Now, if you look below, you can see a schematic of where these electrodes are placed. They're right across from the central sulcus, the CS in this diagram, and they are located in the primary somatosensory cortex, S1, and then across the central sulcus, the primary motor cortex, or M1. And by stimulating lightly fingers on the hand of the monkey, we can get a sense of information travel up to the cortex and across the cortex. And the reason we can do that is because of the homunculus, which you're all familiar with. And you can see the brain, the red strip is the motor strip in the frontal cortex, the blue, the primary somatosensory cortex. And where the electrodes are placed in this monkey brain is in the finger area of the somatosensory cortex. And it wouldn't be surprising that if you were to lightly touch fingers, you can actually decode and classify and predict which finger is being stimulated based on the neural activity in that region. That makes sense. That's why it's a map and a homunculus. What might be less obvious is that you can also analyze neural activity in the primary motor cortex and make inferences about sensory stimulation in the hand. There is a kind of echo representation and a communication between S1 and M1. And of course, this makes sense. If I'm moving my fingers, if I'm motorically active, the brain needs to know where I am in space and what I'm experiencing from a sensory perspective. So I had colleagues in biomedical engineering who were utilizing this paradigm to better optimize brain-machine interfaces, but I saw it as a great opportunity to study drugs because we can use these recordings as a surrogate for thalamocortical information transfer based on what's happening in the sensory cortex, and cortical information transfer based on what's happening in the motor cortex, because primary motor cortex is not directly receiving sensory information. It's coming primarily through S1. So let's look at what happens within this model when we give a monkey ketamine. And this is at doses to make the animal unresponsive. So I want you to focus on the left side of your screen, and for now just focus on the gray bars. And that's reflecting on the y-axis the percentage of correct classification. So my colleagues in biomedical engineering are decoding neural activity from the S1 electrodes, and they're predicting which of 3 fingers on the monkey hand are being stimulated. Now, if I were to predict or get 1 out of 3 right, well, that would be chance, and that's why you see that line at around 33%. So you can see in the baseline pre-drug state that about 90% of the time you can have correct classification. Again, that's what we would expect based on this mapping and the homunculus. But look what happens after ketamine, and again, focus on the gray bars. There appears to be a little drop, although it's not statistically significant, but it is still maintained throughout the exposure of ketamine. And that's really quite interesting. What it suggests is that there is still information transfer from the thalamus to the primary sensory cortex, and perhaps still a high-fidelity representation that could allow us to decode this. Now I want you to focus on the blue bars and look at the pre-drug state. So this is prediction, correct classification from activity in M1, or primary motor cortex. Not as robust as sensory, it's around 70% in baseline, but look what happens after ketamine. It drops down to chance, and it stays around there until the animal starts to recover. And so this suggests that during ketamine anesthesia, you have a preserved thalamocortical information transfer and a disrupted or distorted cortical information transfer. And this isn't just happening because neurons are shutting down. And if you look to the right side of your screen, you can see multi-unit activity, and you can look at it under ketamine in the M1 area, and you're not really seeing a disruption or a depression of neural spike activity. Now, if we move from that level of activity to local field potentials, you can see, if you focus really on the bottom left of your screen, gamma activity is increased in somatosensory cortex. We've heard a lot about gamma today, and I'll be talking a little bit more about that as we get into the human experiments as well. Now, using this same model, this paper was published in 2016 in Neuroimage, and last year we published a follow-up study looking at nitrous oxide exposure. And I won't go through this in detail, but even if you look at monkey W on the left side of your screen, you can see again about 70% correct classification, very similar to the last study I described in the pre-drug state, which drops down to 33% or chance after exposure to nitrous oxide. So a very similar phenomenon. Also similar is the fact that neuronal activity and firing rate is maintained, actually even increased, during exposure to nitrous oxide. So again, this isn't just a trivial depression of neuronal activity that is disrupting representation in the primary motor cortex. And similar to ketamine, if we look at 40 hertz gamma activity, you can see that there is an increase after exposure to nitrous oxide. Now, one difference between nitrous and ketamine is that beta activity also goes up, whereas it is depressed during ketamine. So to summarize this portion of the talk, ketamine and nitrous oxide disrupt cortical-cortical information transfer. It appears that there is preserved thalamocortical sensory information transfer and sensory representation in S1. And this is very consistent with a lot of other studies, including large-scale brain networks and neuroimaging. These alterations are associated with maintained or increased spike activity, so these disruptions are not trivially reducible to a depression of neural activity. And finally, ketamine and nitrous oxide increase gamma oscillations in the cortex. So what does that mean, and where is this going? You have higher frequency activity, you have a disruption of cortical information transfer, you have increase in gamma oscillations that are observed. Well, we might, following Dr. Carhart-Harris's lecture, think about how this contributes to increased complexity. EEG, MEG, magnetoencephalography, and fMRI signal complexity has been shown to roughly track with consciousness. I don't want to oversell anything here, but based on the published literature, if you use the waking state as a frame of reference, complexity is depressed during slow-wave sleep, during seizures, also during anesthesia and coma. It's maintained during REM sleep, and it is increased during the psychedelic experience. And you heard earlier that some of the psychedelic phenomenology is actually correlated with an increase in complexity. And this has been shown for the canonical psychedelics, psilocybin, LSD, and DMT, and also ketamine and nitrous oxide. Now, I think that neurophysiologic complexity might be a useful tool to translate some of the work that we've seen in humans to some of the animal work so we can get at underlying mechanisms. And I'm just going to show you some early directions that we're taking to try to get at more foundational mechanisms. And this is an experimental paradigm in the rodents, so now we're moving from monkeys to rats. And typically it's difficult to assess something like spatiotemporal complexity using the Lempel-Ziv algorithm in rodent experiments because the EEG is typically very sparse. And so there's some other paradigms out there for rodent work, but we developed a high-density EEG montage, and we can modify that, and about 30 channels that you're seeing here. We can modify that by inserting microdialysis catheters in different parts of the brain to measure neurochemistry. In this case, we're going to focus on acetylcholine, as I'll talk to you about in a moment. And then if you look to the right side of your screen, you can see the experimental setup in the chamber, either with the intravenous infusion of ketamine through a central catheter, recording EEG. We had the microdialysis pumps connected in prefrontal and also parietal cortex. And then on the bottom, something quite similar with nitrous oxide. Now, one difference that we just found empirically is that as we start administering nitrous oxide, there's one phase during induction, as we call it in clinical anesthesiology, that appears more like the psychoactive or psychedelic state, but then the animal actually gets depressed behaviorally. And so we're calling that nitrous sedation versus nitrous induction. So let me show you what we found with this paradigm. You can see with ketamine on the top, and this is with normalized limp-Ziv complexity, that during exposure to ketamine, the neurophysiologic complexity increases. And this is something very consistent with what we see in humans. And then it starts to return to baseline in the post-ketamine state. And if you look to the right, you can see a topographic map with the electrodes overlaid and a heat map. So the warmer colors represent an increase relative to the baseline state. And you can see the hotter areas in the prefrontal area and in the posterior area with ketamine. Now with nitrous, looking to the lower part of your screen, again, we have these two states. And during induction, you can see that there's an increase in limp-Ziv complexity. Now note in the topographic map that it's not the same distribution as ketamine. These drugs have distinct mechanisms, although there are a lot of overlapping features. And then during sedation, you see the cooler colors, as we would predict with more of a sedative or anesthetic state that limp-Ziv is going down. And so I think this is encouraging because this is what we see in humans, and we don't really have a good behavioral assay in rodents that's reliable as a marker of a psychedelic experience. Obviously, we can't get a behavioral report. And so I think this might be one promising candidate to explore as a biomarker for psychedelic effects in a rodent model. And this would allow us to start to understand some mechanisms. And so we wanted to start off thinking about some underlying neurochemistry. And this is simplistic and it's just a beginning, but just as complexity seems to track with the capacity for consciousness, so does cortical acetylcholine. It's a very similar pattern where you have a certain level in the waking state. It goes down during non-REM sleep. It goes down during anesthesia, and we've demonstrated this in our laboratory. During REM sleep, it goes up, and there is some early evidence that it might also increase with psychedelic states. And we've shown this for acute exposure to sub-anesthetic ketamine. So we wanted to see, is there any relationship between these changes in complexity and changes in acetylcholine, taking advantage of this model? And what we found is that there is some statistically significant correlation. If you look at the top, that's the prefrontal relationship, both with ketamine on the left, nitrous oxide on the right. Below, in the parietal area, ketamine on the left, and nitrous oxide on the right. Now, I'm not saying that this is all reducible to acetylcholine. Obviously, there's going to be a very complex mosaic of neurochemical events. But I do think that this is interesting to explore, especially given the relationship between complexity, consciousness, and acetylcholine that is manifest in the literature. And what we're doing now is we're starting to explore the underlying mechanisms and modulating cholinergic neurons in the basal forebrain because those neurons supply acetylcholine to the cortex. And what is interesting to think about is whether or not some of the well-known cortical effects of psychedelics are being controlled by subcortical systems. And so that is something that we are currently and actively working on. So to summarize this portion of the talk, the EEG-based effects of psychedelics can be observed across species, and I'm specifically referring to the neurophysiology, although we also see increases in gamma, both 40 hertz and also higher gamma in rodent experiments. Subanesthetic doses of ketamine and nitrous oxide increase spatiotemporal complexity of cortical neurophysiology. And finally, these increases of complexity are correlated with increased acetylcholine. This is correlational, and we have to move to causal studies. And again, there is going to be a very diverse and complex neurochemical mosaic of effects, but we think it's promising to focus on acetylcholine as a start. Okay, now I'm going to move into some of our human work with surgical patients as well as with healthy volunteers, observational studies as well as a randomized controlled trial. And again, Ketamine and nitrous are sitting at this nexus, at the interface between anesthetics and psychedelics. For the remainder of the talk, I'm going to be focusing on ketamine. Again, a very practical way to modulate states of consciousness, psychedelic, and anesthetic in a single experimental protocol. So the first thing I want to show you is some EEG work that was done in human surgical patients. This is low-density EEG. It was in the operating room, although these patients didn't have any pre-medication. And you're looking at 3 classes of drugs, all within the general anesthetic umbrella. Ketamine, which we've talked about, propofol, very commonly used intravenous anesthetic to induce unconsciousness, and also sevoflurane, an inhaled halogenated ether that is used to maintain unconsciousness during a surgical procedure. And you're looking at the relative changes of EEG bandwidths, and we've already heard wonderful introductions to these bandwidths earlier today. Now, what I think is interesting is this combination of canonical anesthetic effects and canonical psychedelic effects on the EEG based on the bandwidth. So, for example, if you look at delta and theta and beta, now the blue traces are what's going down after anesthesia, the red traces are what's going up. So all of these drugs, despite very diverse molecular mechanisms, and especially ketamine differing from propofol and sevoflurane in terms of the system's neuroscience aspects, they have these similar effects in increasing delta and theta, slower wave activity, and reducing beta. So ketamine in this instance is really behaving like the other GABAergic anesthetics. But look what's happening in alpha and gamma. This is where ketamine is differing from its GABAergic counterparts. And instead of increasing alpha, which is a really robust, reliable effect with GABAergic anesthetics at the level of surgical unconsciousness, especially in the anterior part of the brain, ketamine is depressing alpha. And that's something that is consistent across a number of psychedelic drugs. And in fact, the degree of alpha suppression has been correlated with the degree of ego dissolution, and that's subjective phenomenology during a psychedelic experience. And what you can also see, as we saw in the monkey cortex, that gamma activity is getting increased by ketamine, whereas propofol and sevoflurane are depressing it. Now, I want to just touch on alpha a bit because we had another study. This is in healthy volunteers, and these individuals had high-density EEG, and they had a sub-anesthetic infusion of ketamine. The dose is 0.5 milligram per kilogram given over 40 minutes, which is something that's typically used for antidepressant effects. So that was the protocol, 15 healthy volunteers, and you're looking at some source-localized changes in alpha. And actually, our colleague from Liège earlier really kind of foreshadowed this. Look at where alpha seems to be most depressed, and the warmer colors means more alpha suppression. It's in this area of the right temporoparietal junction. And I remember when my postdoctoral fellow presented this and said, "Do you think this is of any interest?" And I said, "I think it's of great interest." Thinking back to the work of Olaf Blanca, and there are a number of studies suggesting this, lesions in the temporoparietal junction can evoke out-of-body experiences. And if you look at the psychedelic phenomenology via the Altered States questionnaire, comparing ketamine to some of the canonical psychedelics, and you've seen a depiction of that during Emma's talk this morning from Studeris et al. 2010, ketamine is really stand out in terms of the sense of disembodiment or dissociation. And in fact, if you think back to the original studies in humans in ketamine, and those first studies were actually done in my department at the University of Michigan in the 1960s. Günter Korsen was an anesthesiologist, and Ed Domino, a clinical pharmacologist. And Dr. Domino went home and described this different state of what was then called CI581, a phencyclidine derivative. And he described it to his wife, this strange disconnection, and she replied, "It sounds like it's a dissociative anesthetic." And so that phrase made it into the title of this article, and it's something that we continue to use today. It's a very reproducible, robust, salient effect of ketamine at subanesthetic levels, and yet nobody's really understood why exactly it causes this sense of dissociation or disembodiment. So I think the TPJ is certainly a candidate that's worthy of exploration, and we've heard about that earlier today in the context of near-death experiences as well. Okay, moving on, I want to just give you a flavor for two articles that we published in Neuroimage, one in 2019, and the other actually is published this month in the April issue of Neuroimage. And this is where we have healthy volunteers who received a subanesthetic dose of ketamine, 0.5 milligram per kilogram given over 40 minutes, as well as an anesthetic dose, 1.5 milligram per kilogram as a single bolus injection. Now, in between those two exposures, the volunteers completed an altered states questionnaire. And we were trying to grasp the various alterations of ketamine, both in the psychedelic and the anesthetized state. And my colleague, Duan Li, who's an engineer with whom I work, again applied limposive complexity, normalized for spectral effects, and used hidden Markov models to determine or define 5 different states. And you can see this depicted here. The y-axis is the spatiotemporal limposive, and you can see the waking consciousness with that center line going across the graph, the psychedelic state above, the anesthetic state below. And you see where they're starting at S1, and then, not unexpectedly, at subanesthetic levels, limposive complexity is increasing. But now look what happens during anesthetic ketamine, because I think this is where the interesting action is taking place. We know that neurophysiologically that there's an alternation early on after an induction dose of ketamine in which your brain is toggling between gamma activity and delta activity. And similarly, this is what's happening with complexity, toggling between a state that would be more akin to waking consciousness, and that's S4, and S3, a state that would likely be more associated with a canonical GABAergic anesthetic. So interesting to think about this ketamine anesthesia state because, as has been noted before, people often report dreams or hallucinations or disconnected conscious experiences. So this toggling and this access to levels of waking consciousness might be contributing to that. And then coming out of the anesthetic state, returning to the subanesthetic levels, again, elevated complexity, and then back to baseline. Now, you've heard from my wonderful colleague, Dr. Zurich Huang, yesterday, who described some of his work that was published in 2020 and 2021 about how functional brain states undergo a kind of temporal circuit. And what you can see with ketamine, and this actually echoes some of what Robin was saying earlier, that a global network is really kind of at the center of this. So each circle represents a functional brain network, and the arrows represent transitions across these brain networks, and the density of the arrows represents the probability of the transition. So you can see all of the heavier arrows in this ketamine where the transition probability is high, all centering around this global network. So now I'm going to depart from the neurobiology and just describe one study related to ketamine and post-operative outcomes in surgical patients. So there have been a lot of positive effects ascribed to ketamine. There was a small study showing that it actually reduced delirium, which is counterintuitive. We wouldn't imagine a psychedelic, either canonical or non-canonical, reducing delirium after surgery, but there was a study of cardiac surgical patients showing that there was a substantive, statistically significant, clinically significant reduction in delirium because of ketamine compared to a placebo, which was associated with a reduction in inflammatory markers. And ketamine is thought to have an anti-inflammatory effect. It's also thought to have an analgesic effect. It's been shown and it's been suggested in meta-analyses to reduce pain scores or reduce opioid consumption after surgery. And certainly a reduction of opioid consumption is important in this day and age with the opioid epidemic. And of course, ketamine is known to have antidepressant effects. And it's been suggested that the administration of ketamine during surgery can improve mood. So I and my colleague, Dr. Michael Avedon from Washington University, he's the chair of anesthesiology there, conducted a multi-center international randomized controlled trial, approximately 670 surgical patients, and they were randomized to either a placebo, 0.5 milligram per kilogram of ketamine, sub-anesthetic dose, after they've already had general anesthesia induced, usually with propofol, so 0.5 mg per kg or 1 mg per kg. Completely blinded, patients are blinded, clinicians were blinded, research investigators were blinded. So a robust study, robust methodology, multi-center, international. And here's what we found. First of all, ketamine did not change the incidence of delirium. No big surprise. I mean, that was a very unique finding, but we thought it was worth exploring because post-operative delirium is a major problem for patients, for families, for health systems, for society. Ketamine has a long-standing safety history. It was worth the risk. We thought, okay, no problem. That was the primary outcome, but let's see what happens with pain and opioid consumption because we know from meta-analyses that this is going to have a positive effect. Well, ketamine didn't change pain scores. In fact, it didn't even change pain scores on the day of the surgery. So there was no acute effect. So, okay. The meta-analysis has shown that it can change pain scores or it can reduce opioid consumption. But it didn't change opioid consumption. And it also didn't improve depressive symptoms. What it did do was increase the incidence of post-operative nightmares and hallucinations. So it didn't do any of the good things that we all were hoping and thinking it was going to do, but it did the bad things that we used to fear. And people have changed their practice with ketamine. People, and we've actually done surveys to assess this formally. They're reluctant to use ketamine in the past because of these psychoactive effects, but then suddenly there was so much positive data and enthusiasm around ketamine, it was getting to that stage of, if we just put it in the water, wouldn't life be better? And I think there's some parallels to be drawn between this work, which is more in the perioperative setting, the surgical setting, and what we see with the classical psychedelics for a variety of psychiatric conditions. Now, I am very enthusiastic about psychedelic research, and I think the global enthusiasm is warranted. I do not think that psychedelic drug research should be stigmatized, but neither should it be romanticized. And I think we have to be rigorous, we have to be methodologically conscientious, because I will tell you, and this is independently of anything neuroscientific, the clinical research literature is littered and strewn with small clinical studies, well designed, but small, single center, that had positive effects and that had dramatically increased effect sizes that all got wiped away with larger multi-center trials. There's a very robust history of this. And again, this has nothing to do with the brain. I'm just talking about clinical trial methodology in general. And conversely, the large clinical trials also reveal adverse outcomes that are not manifest in small initial single-center trials. So again, we should be enthusiastic. I think it's warranted, it's merited, but we also want to be methodologically rigorous, we want to step back, and we want to be critical. And that's one lesson I learned from this clinical trial. So to summarize this part of the talk, ketamine-induced alterations of consciousness have interesting spectral features that are consistent both with canonical anesthetics and also canonical psychedelics, depending on the bandwidth. And they're associated with reduced alpha power in the temporal parietal junction, and that was from source localized data. And in humans, ketamine induces complex changes in neurophysiologic complexity and functional brain network transitions. And finally, based on this clinical trial, administration of psychedelic doses of ketamine during general anesthesia doesn't really have the therapeutic effects we thought it would have, but it does induce some of the adverse effects that we might expect, such as nightmares and hallucinations. So with that, I just want to acknowledge my funding source, the National Institutes of Health. In particular, that top grant was on ketamine and nitrous oxide. And I have to tell you, I had to fight for that grant throughout revisions because the reviewers felt like ketamine and nitrous were so different they should not be studied in the context of anesthetic mechanisms, whereas we thought that's exactly why they should be studied, because if we can understand some fundamental similarities, we can have some mechanistic insight. So I'm glad they finally saw the light. And I also wanted to acknowledge Dr. Edward Domino. Now, I mentioned him as a pioneer in ketamine research. He passed away in the fall. He was an inspiration to us. Now, he died at the age of 96. The month before he died, he published a senior author article in a highly respected journal, okay? And about a week before he died, I got a call from a graduate student who is close with him, telling me that Dr. Domino was in hospice. This was on a Thursday. On Friday, Dr. Domino zoomed in to a course on psychedelic neuroscience I was teaching. He was amazing and so inspiring, so I just wanted to acknowledge him here. Finally, I want to acknowledge all of my colleagues at the Center for Consciousness Science, and this is a core group. This is back when we could have in-person parties. And a lot of the people that did the work are here. Duan Li, Zuri Huang, Michael Brito, who did the rodent work. And we have Dinesh Pal and Anshul Lee and many others who contributed. And finally, I want to thank you for your kind attention, and I'll be happy to address any questions that you might have. Thank you. Now, I'm looking to my colleagues in AV because there were some internet issues earlier. And are we back online for Dr.
Yeah, we're online. We can try.
Okay. Well, why don't I take some questions, and hopefully you can do something magical over there so we can hear Alex and Katrin, because I'm not up for answering questions for the next 90 minutes. Sir.
Yeah, thank you. That was really interesting. I was wondering, you mentioned the increase in acetylcholine that happens in the cortex in relation to ketamine and nitrous oxide administration. And I was wondering if you knew with any greater granularity which cells or which actual parts of the cortex were responsible for that release of acetylcholine.
Yes, great question. I think what we're focusing on is a little bit different. So we're going with the known neuroanatomy. And there are multiple sources for acetylcholine in the brain. One's in the brainstem, but the basal forebrain, which has a heterogeneous population of cells and which contributes to sleep-wake regulation, has a population of cholinergic neurons that project widely to the cortex. And so, the acetylcholine that we're seeing, we think is coming from those cholinergic neurons from the basal forebrain. So what we're doing now is manipulating and modulating and studying those neurons and seeing if there is actually a causal relevance to that acetylcholine release.
Okay, the reason that I ask, and that's really cool you answer it that way, is because in relation to what Dr. Hammeroff has studied with the pyramidal cells in the cortex and the basilar dendrites kind of that span the web, is that the same network, kind of that same pathway that you're mentioning here?
Yeah, I guess I would think about the pyramidal cells as glutamatergic.
Okay.
Obviously there's a diversity of neurochemical mediators, but generally speaking, the cholinergic neurons are going to have an excitatory effect, and as you could, higher acetylcholine is associated with arousal states in the cortex during waking or during REM sleep.
Okay, cool. Thank you so much.
Thank you.
Hey, thank you for the talk. That was amazing. First, wanted to just note that you talked about the TPJ gamma concentration. I'm a big fan of the attention schema theory, Graziano, and he really focuses on TPJ as a focus of consciousness and self. And I don't know, I just think that's a really interesting correlation. I don't know if you've looked into that at all. And then my other question was having talked to some people about some of their like greatest breakthrough individual psychedelic experiences, it is nitrous oxide in combination often with ketamine or LSD or some other powerful psychedelic. And I'm wondering if you've looked at these things in combination as opposed to just on their own.
Yeah, great points and questions. Thank you. So I'm familiar with the attention schema theory. I am by no means an expert, nor do I have a deep knowledge, but I do recognize the role of the temporoparietal junction in that theory. And I think where it's interesting in this context, I've talked about some of the lesion studies, but also other neuroscientific studies suggesting that it's important for body ownership, and it's also a site that's important for multisensory integration, which of course is also important for consciousness. And then your second question was? Yes, the combinations. So yeah, great question. We have not tested that yet. I do think it's interesting when you think about ketamine and nitrous oxide, because oftentimes they're almost considered as interchangeable, as if ketamine is sort of liquid nitrous and nitrous is a gas form of ketamine. They do have a lot of similarities at the molecular level. They work as NMDA antagonists, which comes up a lot, but we also have to keep in mind that HCN1 channels, HCN2 channels are really important. These are like pacemaker channels for the brain. So we haven't studied it in combination, but clinically, I'm sure there are cases where we're giving nitrous oxide, there's a GABAergic anesthetic on board, and then we're also giving ketamine for analgesia. So interesting question. Yes?
Thanks. Very quick question. On your findings that ketamine increased nightmares and hallucinations, did it also increase sleep paralysis? Did you look at that?
Yeah, thank you. So we actually didn't look at that, but that's an interesting question. And ketamine is interesting with respect to sleep because when we think about that metaphor, that analogy between anesthesia and sleep, most of the GABAergic drugs are more akin to a non-REM slow wave sleep state, because, for example, given some of the things I've talked about today, they lower the EEG frequency, they depress acetylcholine, low capacity for experience. Ketamine is sort of more akin to a REM-like state in that you have cortical activation, you have a release of acetylcholine, you have a high incidence of dreaming. So there's an interesting connection, but we didn't study that as an outcome. Stuart, I'm sorry, you've got to get to the back of the line. I can't just let, no, I'm just kidding. Yeah.
The internet is spotty. We have the next speaker sort of online through a cell phone hotspot. This is a nationwide problem, not the hotel. I don't know if it's the Russians or what.
Okay.
But we can try, so I just want to let you know that.
Okay, well, so should we continue with some quick questions?
Then we'll try the next one.
Okay, great.
Okay, thanks. So I'm having this thought, and I guess I just want to check it out with you, because I don't know if it's reasonable, which is that I'm kind of intrigued by this alpha-gamma difference. And it just reminded me of Mark Beeman's work with insight, intuitive responses to compound remote associates problems. Anyway, these problems that you can solve intuitively or analytically. And when you're solving it intuitively, you have this insight, you don't know how you got it, but you solve it correctly. And that's faster than the analytical approach. Anyway, the point is that he found that a depression in alpha and an increase in gamma just before the solution arose to consciousness. And that made me think about, okay, so if these folks are processing, like if they're having insights and they're being more conscious, like sometimes alpha is associated with a bigger consciousness of what's going on with the body, sensory input, and yet you're also kind of sending them out of their bodies. Is the reason they're having nightmares and hallucinations afterwards because they actually saw their surgery, but they don't remember seeing their surgery? And so then I came to this question of, have you analyzed the actual content of the nightmares or the hallucinations to see if there's any associations with what happened during surgery?
Yeah, really interesting. So first of all, in terms of the insight, and you can look back to the work of William James describing, the insights that came from the experiences with nitrous oxide, which, as he said, to paraphrase, seem like nonsense but are really on-sense. And so yes, I think that is certainly consistent with some of that phenomenology. In terms of the awareness piece, it's very rare, and this is something that I've studied. I'll tell you from animal work that we've done, and I didn't present it here, but getting back to what I was saying earlier about this REM-like quality, I've actually studied in animals administration of isoflurane, which is a GABAergic ether, and then adding ketamine. And what I was expecting is, okay, we're going to see the increased cortical acetylcholine and high-frequency activity, and nothing happened. In fact, what happened was the animal got deeper into anesthesia. It went into burst suppression. And this is consistent with an article that was published in Journal of Neuroscience of rabbits who are under isoflurane who got MK801, which is also an NMDA antagonist, and they went into burst suppression. So I think, if anything, the ketamine would induce a deeper state rather than a state of awakening or a lightening of the anesthetic. Thank you. And these three, and then we'll try to move on to the next talk.
This is a question about the GABA receptor that you're postulating being involved in this. Is that the GABA receptor that's influenced by the benzodiazepine class of molecules?
Yes, there are similarities, and they're often considered together as sedative hypnotics. They don't have the exact binding sites, they don't have the same pharmacology, but I think the general effect is similar in terms of that sedation, hypnosis, and by hypnosis I mean sort of disconnection, and you can induce general anesthesia with higher doses of benzodiazepines.
I did research on GABA receptors, and there seems to be, in the allosteric modulation of the receptor itself, you can induce sedation, anxiety, and hypnosis, depending on how you allosterically modulate the receptor. In hypnosis, how would you think that would work with the cholinergic system?
Yeah, I don't know. That's, now I have to admit that I really lied to you at the beginning when I said they work on GABA, because they work on many different things. And it really is all about where the GABA is situated. And that's the systems neuroscience approach. So I don't know if Giancarlo Vanini is in the house here, but he showed that, for example, if you increase GABA in the pontine reticular formation, now normally when you think, I'm going to ramp up GABA, that means everything's going to get depressed. Actually, higher GABA in the pontine reticular formation is associated with wakefulness, and a depression of GABA is associated with anesthesia. Typically, acetylcholine goes down during the administration, and we've studied this in animals, propofol and sevoflurane.
I wonder if you could say a bit more about the out-of-body experience and the TPJ, because in my own experience and reading loads and loads of accounts, there's really quite a big difference between a classic astral projection where you have the body schema is affected, but it's a whole body schema floating around, arms, legs, the whole works. Ketamine, my own experience was of kind of dissolution rather than being be sort of somewhere else, but not with another body, and I think other accounts are rather similar. And then you can have rather in-between ones. Can you say anything from what you've done about the different effects on the TPJ or why this is happening? And I'm kind of wondering whether the system is always trying to get back to a good body schema and failing, and whether that relates to what you've explained to us.
Yeah, those are great points, and we don't have the level of granularity that you're very reasonably requesting.
I want too much.
No, because we're using the Altered States Questionnaire, for example, which has a category of disembodiment. The end. It doesn't get into some of the more granular features. I think in terms of what the experience is going to be like, and I'm sure there are individual factors, there are pharmacogenetic factors, and there are also dose factors. So I had ketamine in a study, actually with Dr. Domino. That picture, actually I forgot to mention that. I just had ketamine, probably why I had that big smile on my face. And you know, I had dissociation. I mean, I couldn't believe the extent of which. But I didn't feel like a full ego death or ego dissolution. So I think, you know, that probably is going to depend on dose. But I think it's a great question. I do think the TPJ is worth investigating more carefully in terms of ketamine's mechanism, because again, we've been using this term dissociative anesthetic since the 1960s, but nobody's ever really shown why exactly it has such a salient effect.
Thank you.
Thank you.
Thank you. First of all, I've been learning so much, so thank you to Abby and Stuart and you for organizing this. My question is related to Suze. Actually, she asked it in a different way, so I'm going to, it's a theoretical question related to TBJ as well. Many times, most people report under psychedelics that, you know, they have like an ego dissolution, as we discussed. But the out-of-body experience, so when I think of ego dissolution, I think it's loss of self-identity in a way, right? But out-of-body experience retains some of that self-identity. So I'd like to just hear your opinion on what are there different parts other than TBJ that are involved with self-identity then?
So let me start with the second question. I have no idea, okay? Now, I haven't studied enough. I think it's a great question. But I do agree with you phenomenologically. In fact, I was having this conversation today with colleagues at lunch, which is the sense of having of being dissociated or feeling like I'm not quite in my body that happens during ketamine is very different than the sense of oceanic boundlessness or a sense of complete unity, ego dissolution, no difference between self and other. I think there's still, at least at some doses, there is still a sense of self with ketamine. It's just that self isn't located in the body. Last question, and then we'll see if we can get Alex Kwan online.
Thank you. I was wondering, what do you think is the connection between the consciousness and complexity and to the cortical acetylcholine?
Yeah, it's a great question. I don't know the answer, and that's really what we're going to try to tease out. And I think it's important to note too, there are many different measures of complexity, and there are different types. And the other thing is, I think, and again, I didn't unpack this, but I think we always have to differentiate between entropy, which can keep going up and up and up, and true complexity, which really is going to have more of an inverted U shape. In terms of, how it proceeds. And so that's something that I think is important for us in the measures. I don't have a good answer for you, though. I mean, I think, I'll resort to kind of a canned answer. When we talk about measures of connectivity, it's often a surrogate for the integration aspect of consciousness. And when we talk about complexity, it's often considered a surrogate for the diversity or the differentiation aspect, but I can't answer beyond that. Again, I'll thank you all, and hopefully we can get Alex Kwan on to now hear about some of the more classical psychedelics. Thank you. Now I'm back up here in moderator mode. And it's my great pleasure to welcome Dr. Alex Kwan from Yale, who has done some really remarkable work in the past few years on psilocybin, and we look forward to his talk.
Okay, well, I hope this works for both the remote audience as well as the in-conference audience. Remotely, sometimes it cut off for 10 seconds or so and then come back, but I hope the connection will be better. I want to thank the organizer for inviting me to give this talk, particularly George. I think it's a very interesting symposium. I'm glad to be part of it. So I'm glad to tell you some of my recent research on psychedelics today. I start with a couple of disclosure. I am an advisor for Empyrean Neuroscience, which is a fungi engineering company, and I have received some investigational drug from Uzona Institute. This includes a psilocybin that I'll tell you about today, but Uzona is a non-profit organization that seeks to advance psychedelic science. That being said, all of the studies that we do are funded by grants. We have an NIMH grant to study ketamine. We have a NASA grant to study some of the earlier work on ketamine. And then most of the psilocybin work is supported by a pilot grant from my home department, which is Yale Psychiatry. This is a research lab. I think I have an amazing lab. I'll try to highlight the contribution of the individual lab member as I go through the presentation. So, we as a lab, we're quite fascinated by psychoactive substances. We are just very curious about how these small molecules can get into the brain and then alter brain function and then eventually lead to some fairly profound changes in behavior. One of our long-term goals is hopefully to be able to bring some of these basic neurobiological insight and mechanisms to be able to start developing circuit models and predict how different other compounds, including novel chemicals, how they might influence the brain and eventually change behavior. So in this context, the three compounds that we have been studying in the labs are ketamine, psilocybin, and 5-MeO-DMT. I will only briefly touch on our work on ketamine. I think George has already done some good work on it, and I will just add a little bit of our own study on that, a couple slides, but to give you a flavor on why we get into this field. And then I'll shift focus on some of our more recent study on psilocybin. I won't talk about the 5-MeO-DMT, but I encourage you to go visit and go listen to this talk in session C6, which is later today, by Robert Cargill from Uzona Institute, who is our collaborator. In fact, have sent us some 5-MeO-DMT, and they have done a lot of exciting work in this area. And then finally, I'll wrap up the talk talking about some newer work that's not published about looking at some more system-wide, brain-wide effects on psilocybin on the mouse brain. So ketamine was discovered as an antidepressant in Yale Psychiatry, again in my department, by John Christo, who's now my department chair. So like others have before him, as well as his own work, he found that if you give people a sub-anesthetic dose of ketamine, it leads to dissociative symptom, actually like what George was saying. Other people refer to these as psychotomimetic symptom, which mimic the positive symptom of schizophrenia. But in general, it leads to these acute behavioral effects that arises really quickly, but then dissipate also quickly within 2 hours or so. But what's more striking, and I think got the attention of psychiatry, the field of psychiatry recently, is these longer lasting behavioral effects. There seems to be like a second phase of behavioral changes that reduces the depressive symptoms. And this eventually led to the approval of the esketamine nasal spray as a treatment for treatment-resistant depression. But what do we know in terms of the neurobiology of ketamine? I would say a lot of that work come from the work of Ron Duman, who was a colleague here at Yale, who was a great scientist and even a better mentor, who sadly passed away a couple of years ago. But Ron and a couple of other people were one of the first to recognize that an important site of action for these drugs, including ketamine, is the dendritic spines. So here, just make sure that we're on the same page. I put on an illustration of a neuron here, a brain cell. You can see the primary neuron, the cell body in the middle, and the very exuberant dendritic tree that comes up. All of the little knobs of the dendritic spines, which are the places where the neurons make their excitatory connections. You can see that piece of red axon from another cell coming in, making that connection. So then the purpose of the dendrite is served to integrate these thousands of inputs, and then the cell body further processes it, and then the neuron can decide whether to fire an action potential to communicate with the next neuron or not. So these are important site of communication for neural circuits. And what Ron has found is that if you stress a rat, then you tend to have a decrease in the number of dendritic spines. But strikingly, if you give the rat now 1 dose of ketamine, you see this restoration of the dendritic spines. This is in the frontal cortex of the animal. So this was quite striking at the time because it showed kind of this structural correlate for what could happen. And this structural change seems to be relatively, it lasts for days, which, consistent with how long ketamine seems to act in humans. So, our own work in ketamine started on a joint lab meeting. We have these every Friday morning. I go there because of the bagel and the coffee, but there's also some good science. And this is when I start learning about Ron's work on ketamine. And, where we come from, I've always been a systems neuroscientist, and my expertise is in live imaging, optical imaging of the live mouse brain. So, Ron showed those results by doing histology and then looking at fixed brain slices and checking the dendritic spines. But we have the capability to look at these things in a live animal. And I'll tell you a little bit more about how we do that. Here you can see an image that we've taken from the brain of a mouse. On the right is a movie showing fluorescence changes. Whenever calcium ion goes into these dendrite, you can see it flashes in the fluorescent increase. So using these kinds of methods, we can look at in a living brain what these drugs do. And we show that ketamine indeed leads to formation of new dendritic spines. So we confirm Ron's work, but in a live brain. We also show that ketamine acutely within that few hours after the administration can increase calcium transient in the dendritic spine. We think these signaling are quite important, could be a precursor for the structural remodeling. And finally, summarizing some of these work last year, we have a review paper reviewing some of these dendritic mechanisms that may explain ketamine's action. So all of this was, I think an explanation of why we started studying these psychoactive substances. And about 3 years ago, I think we decided that, ketamine was quite interesting, but maybe we want to ask if other compounds might exert some similar effects and what do they do in the brain? And this is when we start getting quite curious about various kinds of psychedelics. So the psychedelic that we study in the lab is psilocybin. The chemical is on the left. In the body, it's rapidly metabolized into psilocin, which is what gets into the brain and bind to serotonin receptor that then exert the acute behavioral effect. Both psilocybin and psilocin can be found naturally occurring in the so-called magic mushroom, which is one of the those more than 100 species is taken as a photograph on the right. This is by a famed photographer, Alan Rockefeller. I think for this audience, I don't need to recap what psilocybin does acutely, but it has a variety of intriguing behavioral effects. And but just for completeness, I'll say these passages. This is taken out from T-Call from Shogun and Shogun. These are obviously his personal experiences after consuming psilocybin. And I'll just read them aloud. Visual distortions, things distract me. The furniture in my office is moving up and down. The visuals are wild. I lay down and closed my eyes. It was a South American temple with earth floor, straw, vines full of fruit, familiar feeling. Here today, I feel that everything has disintegrated and I'm extremely anxious. I'm very confused. So I picked these three passages for a number of reasons. I think that because they illustrate different aspects of this acute behavior experience. The first instance illustrate the idea of altered perception. You can see that visual input being modified is a very common consequence. The second passage illustrates the idea of hallucination, which often accompany some of the visual distortions, where you're seeing things that may be imaginary. And then the third passage illustrates the idea that this experience could be positive or negative, and some of this could be due to set and setting, and some of it could also be due to differences in dose dependencies. But what caught our attention, beyond I think the very intriguing acute effect is also some of these longer-lasting potential benefits for treating mental illnesses. And there's a number of work, and I think you all have heard Robin Carhart-Harris' work earlier today. But here I also cite another clinical trial that corroborate those results from Johns Hopkins University. This is a randomized clinical trial. It has two arms and it's waitlist control. So one group received psilocybin early, the other group received psilocybin late. And what you can see is that the group that receives it early showed that reduction in depressive symptom relative to the group that started late. So this, again, with other works, really start to show the promise for psychedelics, particularly psilocybin, for treating depression. And I always forget, so I will also mention, I think unlike medications that are using currently in the market, right? I think psychedelic is quite unique in that the application is in the form of assisted psychotherapy. You don't just give the drug. So I think, you do it in a controlled setting, also guided by one or two medical professionals. So I think that's important to mention. But one of the reasons why we got interested in it too, besides the intriguing acute effect and also the potential benefits for treating mental illnesses is also because of the basic neurobiology. So this is actually a micrograph of the serotonin 2A receptors in the prefrontal cortex in a macaque monkey. I hope you can appreciate how dark that stain is for that receptor in the apical dendrite. These are exactly the dendrite that we have been imaging for ketamine. And you can see that these primary target of the psychedelic is highly, highly enriched in these apical dendrites in the pyramidal neurons in the prefrontal cortex. So this really caught our attention when we started looking for other things to study. When I saw this, I decided, okay, yeah, I mean, these receptors are there. They must be doing something important on the dendrites, and we must study some of these psychedelics. So we don't study the mushroom themselves, but we got the compound from a collaborator at Usona Institute. So again, we don't quite know what we were doing. About 3 years ago, we were just starting. So when we got it, we thought, we should try it on mice though. So this is work by graduate student Clara Liao and postdoc Ian Gregg. So what Clara and Ian did was to start evaluating these psychedelics using a classic behavioral assay in mice, which is known as the Hetwitch response assay. So on the left, hopefully you can see the video of a mouse shaking the head. This is taken by a high-speed camera, overhead camera, and then slow down for you to see is they shake their head about 90 times a second, this movement. It happens spontaneously after you inject psilocybin into the animal. And then on the right is a control animal. So Clara and Ian did a very careful dose dependence study mapping the out the dosage. Consistent with some earlier work, we also find that 1 mg per kg is about a dose where you see this large inflection point where you really see the Hetwitch response to kick in, which has usually been taken as an indicator of hallucinogenic potential in human. So this is the dose that we use for the subsequent study, 1 mg per kg. We were wondering whether this dose also affects some other behavior, particularly behavior that we can measure out further out, because we're interested in some of the beneficial action that are longer lasting. So we decided to do a task called learned helplessness, which is a stress-related paradigm for mice. This task started with 2 days of inescapable shock where the mice receive foot shock, but they cannot escape it. Then there's a test section where the mouse could shuttle and go to the other side of the chamber, which would stop the shock. But as the name of the task imply, a lot of the mice learn to be helpless because of the initial shock. And then after the first test day, we would give the mice psilocybin, ketamine, or saline, and then we would test it again to see what happens. So what we found is that a fraction of the mice would escape and some of them will not. And then here on the y-axis, I show you just a failure to escape. You can see that when we inject the mice with saline, it didn't change that much. But when we inject with ketamine, there's a decrease in escape failure, meaning the mice could escape more. So they seem to be less sensitive to that earlier episode of uncontrollable stress. Ketamine, we tried that too, also seemed to decrease the level, although actually the effect was a bit less than what we see with psilocybin. So this suggests that even a day later, when the drug is fully out of the body of the mouse, you can still see some behavioral effect, kind of consistent with what some of these pilot clinical trials show in humans. But as I mentioned, we are a systems neuroscience lab, so we were very interested in what the drug does inside the brain. So we apply some of these imaging methods, and this is how we do it. So on the left is a micrograph of a transgenic mouse where the neuron expresses a fluorescent protein, so they were lit up under our microscope. All of our imaging is done with live mouse, so they're under a so-called 2-photon microscope that allow us to image neurons in subcellular resolution. We're imaging in the prefrontal cortex, which is the front of the mouse, which relates to probably the anterior cingulate cortex in human. And finally, to image it, we typically do a craniotomy on the head, and then we would replace it with a piece of glass window for optical transmission. So if you were to come to my lab at Yale, this is the kind of image that you might see under our microscope. Here we're imaging those Taiwan GFP mice, and I'm going into the brain. You can hopefully see the dendrites coming in as I go into the brain. And hopefully you can also see the little knobs, which again are the dendritic spines, where a lot of the excitatory connections are made onto the neurons. So here again, we can visualize these in a live mouse. The postdoc who's leading the work is Ling Xiao, and she's getting exceedingly good at doing these kinds of imaging. And we can get it sparse where we can just image 1 or 2 cells at a time even. And this is now a side-on view of a neuron that we can image in a live mouse brain. Again, you can see the very elaborate dendritic tree that comes up, including the spines and everything. Especially you can see how it fan out on layer 1, which is where it receives a lot of the cortical inputs, the long-range inputs for these neurons. So Ling Xiao decided to ask, what is the effect of psilocybin on some of these dendritic architecture? She would do a surgery, image for 2 times to establish a baseline, give the animal a psilocybin or saline, and then image a number more times, and then finally one last time about a month later. To show you the quality of the data, here's the kind of images that Ling Xiao could get. Hope you can appreciate the stability of it, how we can really track the same piece of neuron in the same dendritic segment over many days repeatedly. We can really see those kind of neuronal connections, including some of the new spines that comes up, some of the stable spines. Many of these connections are stable, and then also some spines that are actually eliminated over time. So I think the main finding that we have for this study is that quite strikingly, psilocybin increases the spine density in the brain and also does so in a very enduring manner, very long-lasting effect. So here you can see we're counting spine density, which is really just the number of spines on each segment. You can see a 10% increase on the day after the psilocybin has been administered relative to saline. And this effect was sustained, and we can even see it out to day 34, which is a month of time, which is, again, very long-lasting. We look at both female and male animal, and a little bit to our surprise, we didn't intend to find any sex difference, but we did observe a bigger effect in female. We have imaged this with a complementary method called Confocal Microscopy that showed a similar result, so we're quite confident about this sex difference. But it does seem like the psilocybin has a strong effect on female in terms of inducing the structural remodeling in the animals. We also look at the size of the spine, which reflect the strength of that synaptic contact. So the bigger spines are stronger and they can transmit more electrical signals to the neurons. Here we see that psilocybin also has a pretty strong effect of about 10% increase in the spine size immediately, although it's not as long lasting. You can see it go back to baseline by 1 month's time. You can see it gradually dropping. And this also seemed to have a sex difference where female also have a bigger change. One of the key questions that we have was, what are some of the mechanisms for these processes? It's very exciting to see the spine increase over a month because, again, a long-lasting, not very many things in the brain is such long-lasting, and it really corresponds, I think, well to some of the benefits that people see in humans. But what might be the mechanism for this process? And I think one of the first questions that we have was whether the 5-HT2A receptor, the serotonin receptors, are important because many people have shown already that the 2A receptors, including some of Katrin's work, who talked after me, show that this receptor is critical for mediating the psychedelic action in humans. So whether these same receptor might be responsible for the structural remodeling we see is a big question for us. So the way we addressed this was to pre-treat the animal with ketanserin, which is a 2A receptor blocker, which would reduce the number of functional receptors, and then give the animal psilocybin. So indeed, when we do this, we see that it completely abolishes the behavior. The animal no longer does the Hetwitch behavior. It's just completely gone. And then what Ling Xiao did then is to repeat some of those spine imaging experiments, but instead of giving just psilocybin and saline, she would also pre-treat the animal with ketanserin this time. And what she found was that this pre-treatment really didn't do much to the structural remodeling. You can see that there's still an effect for the spine density, even though it's not significant. You can still see quite clearly there's an effect. And then there was a significant increase for the spine head width and also for the formation rate, which is really just the day-to-day turnover of these spines. So all in all, the data, the way we interpret it is that the ketanserin really didn't do much, and it seems like the structural remodeling could proceed even though we're trying to block the 2A receptor. Which is quite intriguing because it suggests that perhaps some of the other receptor that psilocybin bind to, maybe 2C receptor, 1A receptor, some of those other receptors could also be influencing the structural remodeling process. One caveat of this experiment, though, is that the ketanserin, at least in the rodents, do not block all the 2A receptors. They block about 30% of them. So there are some remaining functional receptors that could still potentially mediate these processes. So I want to take a step back now and just to summarize this segment. What does it mean to have more spines, and why might we want to see this? I think one more biological explanation is that there has been a lot of work showing synaptic atrophy in depression. So on the left is a PET study where you can use a synaptic tracer to ask what happens comparing a control human subject with a depressed human subject. And you can see a loss of synaptic material in people with depression. So one possibility is that this increase in the dendritic spine that we see might be just really good in terms of restoring some of those synaptic connectivity that has been lost. Another possibility that I am quite fond of is that it could also facilitate a long-term integration of new experiences. Here, you could ask, when do you usually see spine formation in an animal? In adult, most of the neuronal connections are relatively fixed. You have certain skills and certain memories, and you might not want to lose them. So there's a couple of papers about 10 years ago now showing that in an adult animal, the time when you see a spine forming is when they learn something. So here, these researchers did an experiment where they have the mice reach for a piece of spaghetti. And in learning that motor action, they can see the new spines forming in their motor cortex. So I think, when we see these spines being formed in the frontal cortex due to psilocybin, it might also be a reflection of just the brain being a bit more malleable, a bit more plastic, being more receptive to some of the new experiences. And I think there's some, we can speculate it, but I think this is also, the exact time when you have these acute experiences and the assisted psychotherapy. So again, I think this is a more, subjective experience-dependent type of explanation for how this happens. I think I already kind of said this, but I think it's interesting to think about what this might mean. I think the spine density could be interpreted both fairly biologically in terms of restoring the spine density, but also matches well with this framework that the subjective effects of the psychedelics may be necessary for some of the therapeutic effects. I think the plasticity in general is just quite important, and it just reflects the brain being able to change, and and the fact that these drugs can do it for long periods is very intriguing. So one last piece of study I want to share with you is some ongoing work that's unpublished. This is work done by MD PhD student Pasha Davodian. So most of the work I show you, and actually I would say most of the work in the field has been really focused on cortex and a few other areas like the hippocampus. But presumably these drugs compounds are administered systemically, right? So everywhere in the brain would receive these compounds. So what other brain region might be involved in the action of these psychedelic drugs? So what Pasha did was to do a whole brain study. So he would give animals either psilocybin, ketamine, or saline. And then we're looking at the acute time point where these drugs are still active and bioavailable. He would collect the brain and then do a brain clearing method and then use light sheet microscopy to image the whole brain. So how that looks like is something like this. On the left is just a segment of this light sheet. So again, the brain is completely clear, right? And we're imaging with a light sheet, so you can see a sheet of the brain. And the fluorescence is we're immunostaining. We're staining for this protein called CFOS, which is a plasticity-related transcription factor. And we're guessing that this protein, because it's heavily involved in plasticity, probably get invoked when the psychedelic is being administered and is involved in that subsequent structural remodeling that we see. So again, we can now, basically what it's saying is we look at these changes in the CFOS expression across the whole mouse brain. So I don't expect you to be able to see the detail in this plot, but I just want to show you just the detail in terms of the large scale data set that we can obtain with this method. We basically match the data we get with the Allen Brain Institute's Brain Atlas, which has over 300 different regions divided in the mouse brain. And we, again, administer both ketamine and psilocybin, so we can see on a region by region basis what the changes in terms of the CFOS expression after administering these drugs. But just to summarize that data, here I pick out some of the more salient things that we have observed. On the x-axis is a change due to psilocybin. You can either have an increase or decrease in CFOS. Y-axis is ketamine. So what jump out at us is that some of the regions have shared targets for both of these compounds. So for example, ACAD, which is the anterior cingulate cortex, is one of the, one on the top right quadrant. And that's exactly the place where we're doing some of the spine imaging. So it seems to be commonly targeted by both ketamine and psilocybin. LC is the locus coeruleus, which is releasing the norepinephrine, which also relate to arousal. VISP is the primary visual cortex. But you also have some areas that are selectively targeted by psilocybin, which I think are quite fascinating and places we would love to do more study on. For example, LH is the lateral habenula. which has been implicated in a lot of stress-related studies. And then AID is the anterior insular cortex, which I think is really understudied, and it has a lot of I think work implicating it for emotional processing, but it's understudied in, I think, systems neuroscience. And there are also some ketamine-specific areas. On the y-axis, you might see the RSP, which is retrosplenial cortex. There was a study from Stanford University a couple of years ago showing that some of these dissociative effects of ketamine might come from the retrosplenial cortex, and that also showed up in our brain-wide screen. One of the things that we can do with this kind of data is comparing to some of the bioinformatics that's out there. So this actually, we borrow a page from some of the fMRI study, again, done by Katrin and also some of our colleague at Yale Psychiatry, where you can relate now these whole brain CFOS map that we have with some of the known gene expression map that's also region by region across the whole brain. So here we took all the gene within the mouse genome and then correlate, again, on a region by region basis with some of these CFOS changes that we observe as a way to just explore what might be some of the genes that underlie the transcriptional changes, plasticity changes that we might observe. On the left is a plot for ketamine, on the right is a plot for psilocybin. To the right means that it's more correlated. I think the surprise here is that the glutamate receptors are really the shared target for these two compounds. For ketamine, maybe it's not that surprising, and it's an NMDA receptor antagonist. But for psilocybin, there's also seemed to be this high correlation between the glutamate receptor expression with its effect on the CFOS, suggesting that some of these excitatory pyramidal neurons might be quite important for the drug's effect on plasticity. The other thing that's comforting to see, but maybe not surprising, is that for psilocybin, you can see the HTR2A, which is a serotonin 2A receptor, correlate quite well for psilocybin, but not so much for ketamine, which is what one might expect here. So to summarize all of this work, I show you some evidence that psilocybin can evoke and boost structural neuroplasticity. Again, I think here the surprise is that it's just such long-lasting effect, which matches quite well with some of the benefits that people see, which are also very enduring. We think that details matter, like where does plasticity occur? That's why we're doing some of those CFOS study. And also when is that plasticity being boosted? Because I think knowing the place and also the time will be quite important if we want to augment this process. If there's any other thing we can do to try to further enhance the plasticity, that would be great. And finally, I think we're quite keen on understanding, again, these drugs, systemic administration, they should be affecting the whole brain. But why is it that certain regions seems to be more sensitive than others? We're very keen on understanding some of the circuit mechanism that might shape the effects of psychedelics. So with that, I hope you enjoyed the presentation. I hope the connection is okay, and I'm happy to take any questions.
Thank you very much, Alex, for that elegant talk, and we have some questions.
Hi, Alex. Here's Christoph Koch from the Allen Institute. So, can you say something about the claustrum? I noticed in you have some you have some C4 expression in the claustrum for both ketamine and as well as psilocybin. Yeah, maybe that's.
Yeah. Well, first of all, I want to thank you, Christoph, for the gene expression map and all of those databases from the Allen Brain Institute.
You're welcome.
And then secondly, yes, we put that claustrum down, I think, because of the interest. I think just because in general for consciousness, but also I think there's been suggestions, for example, from David Nichols. Again, there's just a very high density of 2A receptors. And there are also some recent very good work from Johns Hopkins fMRI showing some connectivity change with claustrum. Yeah, so when we look at it, there is a significant change in the CFOS expression in the claustrum. Seems to be true for both psilocybin and ketamine. I don't have anything more to say than that, besides the fact that we looked at it and it increased. But yeah, it's quite interesting to see that it pops up as one of the regions in this kind of unbiased screen.
Okay, and the second question I have is sort of an obvious one. Have you ever done, giving the animal psilocybin and then actually observing just with a, either two photon or single photon activity, using some GCAM?
Yeah, so that's an ongoing study. So with the ketamine, as I show you just that snippet of the movie, we were very interested in dendritic signals, like functional signal that might underlie the mechanism. So I want to understand how. So we have been doing some of that with psilocybin as well. I think that's a very natural question. We also started looking at different cell types. So the pyramidal tract neuron, PT neuron, IT neurons. So what I can tell you is when we look at the PT neuron specifically, and we look at synaptic calcium transient in the dendritic spine, we see very similar to ketamine. We also see a notable increase in the synaptic calcium transient going into the spines when we administer a awake mouse with psilocybin. Suggesting, again, this calcium influx might be general across ketamine and psilocybin, which I think is quite interesting. Again, I mean, the 2A receptor is out in the dendrite. Supposedly it has an excitatory influence, but to see it and to see it have an effect was quite comforting. Yeah. But we're still doing a bit more. Yeah, so good.
Stuart?
Thanks, Alex. That was very interesting. Since ketanserin blocked to some extent, at least, and ketamine mostly comes through a different receptor, is it possible? And since the cytoskeleton is essential for neuroplasticity, including dendritic spine formation, the microtubules actually go in, and then actin fills it up. These drugs are nonpolar and most likely do get inside the cell. Is it possible that they're acting directly on the cytoskeleton to promote plasticity?
It's possible, I think, but that's I think maybe downstream to our processes. I mean, I think when we think of these drugs, we think of their action on the NMDA receptor and the serotonin receptor, just because that's where they bind. And then the next step is we think it might act on some neurotrophic factor. But eventually downstream, I agree. It definitely goes into activity-dependent transcription. To build a synapse, you need microtubule, you need synaptic vesicle on the presynaptic side, you need other postsynaptic density. And those would be the subsequent process of building out the synapses. But we haven't looked at that explicit link. I mean, I think, we're just at the first step.
Thank you.
And a final question for Dr. Kwan.
Yeah, great talk. I actually am very curious about the question that was just asked, and it segues a bit into what I wanted to ask. And, given the plasticity effects here, do you think there's any efficacy in using this in something like helping patients with cerebral palsy sort of repair their brain, or even looking at patients with something like hereditary spastic paraplegia, where there are alterations in dendritic spines, but it's actually due to issues with microtubule severing. And so I was wondering, do you think there's any efficacy in looking at experiments like that, either in animal or human models?
Yeah, I think the fact that these compounds like ketamine, or particularly the psilocybin, can boost plasticity makes them very interesting as a treatment option. I think it may be why they could be applicable to a range of disorders as opposed to just one. But the one I'm thinking of actually in my mind is substance use disorder. I think there's also good evidence there from a number of groups and also clinical trial, for example, for tobacco use disorder or nicotine disorder, that also seem to be quite responsive to psychedelic. No, so I think there is definitely potential. Again, I think that's why I also raised the question, where and when the plasticity occur, and whether I think you can augment some of these. I think being able to target that and being able to bring that to where you need it will be quite useful if we can figure that out. But I think it could be potentially a bit more broad than just a few disorders. But I think we need to know what's going on first.
Thank you.
How about a round of applause for Alex Kwan? Alex, thank you very much, and we apologize for the internet issues. And now we have the pleasure of Dr. Katrin Preller from the University of Zurich, who has been doing some fantastic work, has presented at these conferences in the past. And Katrin, in addition to apologizing for the internet issues, I do need to apologize for the time difference. I know it is very late right now in Switzerland. So thank you so much for sticking with us. And on to Dr. Preller.
Thank you so much. I hope you can hear me well. If not, please interrupt me. And it's a pleasure to be here again. Unfortunately, this time not in person, but I hope next year I can join in person again. It's a pleasure to talk about the neurobiology of altered states of consciousness today. And hang on. So as the previous talks, I'm going to focus mainly on altered states of consciousness induced by psychedelic substances. So why do we consider the neurobiology of these substances to be important? Well, as previous talks already outlined, these substances induce an altered state of consciousness, which is characterized by many things, including visual alterations and alterations in body perception and self-perception. So that, first of all, means that using these substances is a unique opportunity to better understand the brain and how the brain creates these unique effects. And then on the other hand, of course, we have seen in these preliminary trials that psychedelics can help patients across different diagnoses. So of course, we want to understand why this is the case and how these substances help these patients to be able to eventually optimize the therapeutic approach. The two substances that my talk will cover are psilocybin and LSD, and you already heard quite a bit about them. So they are preferential serotonin 1A and 2A agonists in the case of psilocybin. LSD has a bit of a more diverse pharmacology, stimulating various serotonin and dopamine receptors. And both of these substances induce a pretty similar altered state of consciousness, as we have seen in previous presentations already. Now, luckily, Alex has already introduced catanserin. So what is similar across these two substances is that in the humans, catanserin blocks the serotonin 2A receptor. And if we administer psilocybin or LSD after we have administered catanserin, our participants basically do not experience any kind of altered state of consciousness. In fact, it's really, if we administer catanserin first, it's not distinguishable for our participants whether they have received placebo or catanserin plus LSD. And this is true for psilocybin and LSD, at least at these doses that it was administered here, where we usually use moderate doses, which elicit this very clear altered state of consciousness, but it's not a dose where participants do not know anymore where they are, for example. So this shows us that the serotonin 2A receptor is critical for these subjective effects for creating this altered state of consciousness. Now, we know psychedelics target the serotonin 2A receptor, but how do we get from the serotonin 2A receptor to this altered state of consciousness? And then from targeting the receptor and this altered state of consciousness to the clinical effects that we see in the studies that have been published recently? Well, first of all, when a psychedelic reaches the brain and stimulates the serotonin 2A receptor, we know that the serotonin 2A receptor is a excitatory neuroreceptor, so it increases the likelihood that the neuron becomes active. But there are a couple of things that influences whether the neuron becomes active or is more likely to become active. First of all, serotonin 2A receptors are expressed throughout the whole brain, but in some brain areas they're expressed more than in others. So what happens in the brain will likely depend on where exactly the serotonin receptors are located and which brain area is then stimulated more or less. Of course, the effects also depend on the dose that is being administered and the exact substance, because as I said in the beginning, even though all of these classic psychedelics target the serotonin 2A receptor, there are other receptors involved as well, which means that they probably have a modulating effect and therefore are also accountable for the slightly different effects of all these substances. Now, if a neuron is more likely to become active, that also means that the information that is processed in the brain is different, because the way that the information is passed from one brain area to the other is going to be different if the neurons are activated in a different way. So, and if information processing within the brain is different, that also means that it's very likely that our brain will interact differently with the body and with the environment. So in the first part of my talk, I'm going to focus on just the brain itself. And what we do is, when we just want to study how information processing is happening in the brain while it's not interacting with the environment, we usually use resting state. So we put people in a brain scanner and we just look at what the brain is doing while it's not being challenged with any task or anything else. So to explain the results that I'm going to show you in just a minute, I just quickly want to go over how our brain is working. So we have different brain areas, and there is a differentiation between sensory brain areas and association brain areas. And the sensory brain areas are the ones you see in color here. So for example, here the back of our brain is responsible for receiving visual input and processing visual stimuli. The gray areas that you see here, these are associative brain regions. Now, these brain regions are responsible for integrating all this sensory input and acting upon this input. So making plans or connecting that with past events and so on. So keep that picture in mind because it's going to be helpful for interpreting the brain scans that I'm about to show you. So the first hypothesis that we wanted to test was on the thalamic gating model. So it was suggested based on animal studies that a region in the middle of our brain, the so-called thalamus, is not working as well as it usually does under the influence of a psychedelic. Now, what the thalamus does is it is filtering the information that reaches our brain, and this information which reaches the rest of the brain, the cortex, is then processed further. Now, it was hypothesized that under the influence of a psychedelic, this filtering function is reduced. So more information is reaching the cortex, and this leads to a sensory overload, which is then experienced as the psychedelic symptoms that our participants report. Now, to test this model, again, we administered LSD in that case. We put our participants in the scanner, and we looked at their functional connectivity. So we looked at how is the thalamus interacting with the rest of the brain. And what you can see in warm colors is increased connectivity, and in cool colors, you see decreased connectivity. So if you still have that picture in mind that I showed in the beginning, you can clearly see that the thalamus is interacting more strongly with the sensory brain regions, but is interacting less with these associative brain regions. However, there is a downside to using functional connectivity here, because functional connectivity can go both ways. So we don't know whether there's an increased information flow from the sensory brain regions to the thalamus or the other way around, which is what is hypothesized in the thalamic filter model. So to be able to test this, we had to use a second technique, which is called dynamic causal modeling. Dynamic causal modeling can give us insight into the directionality of these effects. However, at least for the time being, it's hard to really study the whole brain, so we have to focus on specific regions of interest. And what we did here is pick specific regions of interest, which are key regions for the model we were testing, and that is the ventral striatum, the thalamus, of course, which is the core of the theory, the temporal cortex, and the posterior cingulate cortex. And what we see here is that indeed the thalamus has a stronger connectivity to the posterior cingulate cortex. It also has weaker connectivity to some other brain areas in the cortex. But in general, that shows that, yes, there is increased connectivity from the thalamus to certain cortical brain regions. So again, same picture, keep that in mind for the next few slides as well. Differentiation between sensory and associative brain regions. because we were not only interested in what the thalamus is doing and how the thalamus contributes to psychedelic effects, we were of course also interested in what happens after the thalamus has reduced its filtering, the information is in the cortex. And what you can see here is LSD versus placebo and psilocybin versus placebo. And strikingly, both substances induce pretty similar effects in the brain. But to dive a little bit deeper, what you see here is a measure called global functional connectivity. So it's representing the connectivity between each voxel and the rest of the brain. And you see a striking differentiation between sensory and associative brain networks here, with increases in connectivity between sensory brain regions, again, the warm colors, again, remember the visual cortex here at the back of the brain, while at the same time, our association brain regions are disconnected, or at least less connected to the rest of the brain, under both LSD and psilocybin. Now, what does that mean? Well, first of all, to bring these two results together, we have seen that the thalamic filtering is reduced, leading to increased connectivity, particularly to sensory brain regions. And at the same time, we see heightened sensory processing indicated by increased connectivity between sensory brain regions, which is not counterbalanced by associative processing. And that reflects really nicely the state that our participants report, which is usually a highly sensory state, but the way we integrate this information is different. And that could explain, for example, why our sensory experiences in general are different, because we integrate this information different and because we have this heightened state. But it also could explain why patients develop a new view on themselves and the world, because integrating information differently may also lead to be able to see themselves in a new way and to perceive the world differently, because we bring the information together in a different way and connect them differently. Now, we were also interested if we could predict these changes in connectivity, because of course, eventually it would be great to have a measure that could tell us how someone is going to respond to the treatment or react to the substance before we even administer the substance. And here we saw that the functional connectivity, so the functional architecture of the brain without any substance on board, is predictive of how the brain, or the magnitude of how the brain reacts to the psychedelic substance, meaning that our baseline state, our regular state, seems to have something to do with how the brain then reacts to psilocybin in that case. That is obviously not yet a biomarker for treatment response, but it needs to be tested in clinical studies to see whether indeed we can predict treatment efficacy from the baseline state that our brain is in. And this is something we're testing in clinical studies right now. Of course, we were also interested in how specific these effects are for pharmacologically induced altered states of consciousness versus non-pharmacologically induced altered states of consciousness. And this is a project really spearheaded by two PhD students, Flora Moraes and Nathalie Riese, who looked at psilocybin versus a control condition, placebo in that case, and LSD versus control, as well as hypnosis and meditation. And as you can see pretty easily, is that while there is some overlap between psilocybin and LSD, and I'll come to that in just a second, but the state seems to be very different when we compare that to hypnosis and when we compare that to medication. So it seems like there is something else that is going on in these pharmacologically induced altered states of consciousness compared to hypnosis and meditation. When we compare psilocybin directly with LSD, we indeed do not see any statistical differences between these two states. which is kind of what we saw in the previous slides as well, where we just looked at it and saw by eye that they look very similar. And even when we do the statistical comparison, we don't see any changes with that data-driven approach that we used here. We also conducted, trained a classifier to differentiate between these different states. And this is basically also revealing the same picture that we already saw by eye when we looked at these maps. And what we see here is that, you know, LSD and psilocybin basically don't get confused with meditation and hypnosis. And the same holds true for the non-pharmacologically induced altered states of consciousness. So they don't get confused with LSD or psilocybin, at least not that much. However, when we only look at LSD and psilocybin, we see that the classifier is absolutely not able to differentiate whether someone has received psilocybin or LSD. But the picture becomes a little bit more diverse with meditation and hypnosis. Again, there is definitely more confusion going on than between, for example, meditation and LSD. However, the classifier is doing quite well to differentiate between hypnosis and meditation. So our psychedelics, our classic psychedelics, induce a pretty distinct state of consciousness in terms of its neurocorrelates. But it seems like meditation and hypnosis are doing something different, also something different from each other. And I think that this is actually pretty interesting, because that could also mean that hypnosis and meditation, maybe in combination with psilocybin and LSD, might have synergistic effects and complement each other. So the second part of my talk is going to be about the interaction between the brain and the body and the environment. So as I said before, we have now seen that information is processed differently under the influence of a psychedelic. If information is processed differently, it will certainly interact differently with the environment. And one thing that we were particularly interested is how the brain produces these changes in bodily self-perception. And again, another project spearheaded by a PhD student, and Patricia Dürler, she tested a tactile mismatch negativity paradigm under the influence of psilocybin. And what that means is that we put electrodes on the forearm of our participants. And these electrodes, they were not inducing any shock or anything. It was just more of a tickling sensation. But we gave our participants the tiny little shocks at a particular intensity, and we provided them in a row. So we had the same intensity for about 3 to 7 times. And usually when the brain receives the same input over and over, it habituates to that input, and it decreases its reaction to this input. And then after a while, we change the intensity, which is then a new stimulus for the brain, which usually creates a surprise reaction, which is reflected in the EEG signal as this mismatch negativity. And a habituated stimulus is the S here. So that's the second or third stimulus in a row, or the third or fourth stimulus in a row. And the new stimulus, the surprising stimulus, is the so-called deviant. So we did that while we scanned our participants again in the fMRI and also conducted EEG simultaneously. And these are the results here. So what you see here is always the difference between the deviant, so the surprising stimulus, and the habituated stimulus. And usually you see a strong reaction of the brain when it receives this new, the surprising stimulus. And here we see that psilocybin attenuates this reaction to this new, this novel stimulus, especially in frontal brain regions. And when we look at our EEG data, again, down here, we see that we have this, in the placebo condition, we have the significant difference between the new stimulus, the surprising stimulus, and the habituated stimulus. And this, again, as in the fMRI data, we see that this is not the case when we administer psilocybin. So the mismatch negativity is reduced, again, over frontal electrodes. And if we look at these maps up here, we see, again, the increased response to the deviant, to the novel stimulus. And under psilocybin, it seems that the standard, so the repeated stimulus, is becoming something like the deviant. So it's processing these habituated stimuli as if they were novel stimuli all over again. And when we correlate these results, this reduction in mismatch negativity with what our participants are experiencing, we see that this signal correlates pretty strongly with disembodiment induced by psilocybin and by experience of unity, again, induced by psilocybin. So the bodily alterations that our participants are reporting. So the last part, and I will keep that part short, is about social interaction, because of course, the brain does not just interact with the body, but it also interacts with our general environment. And of course, our social environment is a particularly important one. And I'm just going to show the summary figure here, because what I want to point out is that psychedelics are also a very potent modulator of social cognition, of social perception, and social interaction. And this may also be one of the drivers of clinical efficacy, because we know that a lot of patients have deficits in, for example, empathy processing. They show social withdrawal. And of course, also the therapeutic process, the interaction with the therapist, is a social one. And modulating social cognition could be one of the effects that drive clinical efficacy. And with that, I want to sum it up. So we have seen that increased bottom-up processing together with this altered information integration may underlie the psychedelic state. We have seen this potential hypersensitivity to incoming stimuli and a reduction in forming memory traces. So hypersensitivity potentially driven by the thalamic cortical connections, and a reduction in forming memory traces because the the regular stimulus, the habituated stimulus, is being processed just like a new stimulus. We have seen that these changes in processing of novel tactile information may underlie the psychedelic-induced alterations of bodily awareness, which has been shown in these correlations, for example, with experience of unity. And these patterns of altered information processing and integration may actually help to escape ruminative thinking patterns and provide this novel perspective on the self, which may be particularly important when we think about therapeutic effects. However, it has to be noted that all of these are hypotheses when it comes to the clinical response and need to be tested in clinical studies. And this is something we're working on right now. And I hope when I come back next time, I will have more new data to talk about how all of this relates to clinical effects. And with that, I want to thank everyone involved and of course you for your attention.
Katrin, thank you so much for that wonderful talk. Any scientist can give a great talk, but to give one at 1 a.m. European time really requires somebody exceptional. So thank you so much. Yes, another round of applause for that. Do we have any questions for Dr. Preller? Please.
Hey there, I just have a quick one about when you're talking about the meditation, comparing meditation with the psychedelic states. What specifically was the meditation instruction? Because I know there's a lot of variation there and how that can actually be measured or quantized.
Yeah, so here we looked at open meditation. Yeah, so it was open meditation, so basically just focusing on breathing, but no specific instructions other than that. However, I have to say that these were experienced meditators, so they were not people who did not know how to meditate. They had hundreds of hours of meditation practice and knew what they were doing. And we also checked whether they were able to meditate in the scanner and have ratings for that. And because they were such experienced meditators, that actually worked really well.
Cool, thanks.
Katrin, I have a question for you. I thought your data on the mismatch negativity was really fascinating. I was wondering what you think is going on. Do you think that, looking at it from a predictive coding standpoint, that psychedelics are disrupting priors and really changing expectations? What are your thoughts on that?
Yeah, I mean, that's such an interesting question, and I wish I had a good answer to that. Unfortunately, it's kind of hard to tell from these data because we don't have any measure of top-down versus bottom-up processing really. So yeah, so I don't think we can really tell from these data whether it's the increased sensory input or increased bottom-up information flow, or whether it's really a disruption of top-down priors, or whether it's both at the same time. I think for that we need other studies to be able to really tell.
Thank you. And it looks like this is the end of our session. Katrin, again, thank you so much for sticking with us. We're sorry about the internet issues. And how about one more round of applause for Dr. Preller? Thank you all very, very much. Enjoy the evening.